Good morning and welcome everyone. Today I will give the road towards biotechnology. I will start by the National Center for Biotechnology information. Here I have prepared a short presentation for you to know more about the Ncbi and how it works. It is one of the largest database found worldwide. You can see the first thing and even the most important thing that most of us know is the PubMed. The PubMed is a service that allows you to search for keywords in research and publications, and you can filter the results by many filters such as here. I will start to show you how to use the PubMed. I will click on the PubMed. I will write my research keywords, for example insulin. As you can see here, it gives you a lot of results which is more than 360,000 here. I may use filters to minimize these results, to go through a small number which I can skim and scan firstly to select the research topics or articles that I need to read. Here, for example, in the filters is the article type. I can choose the article type that mostly suits me. Here is clinical trials, reviews, systematic review. You can even click on customize to find more, such as guidelines, governmental publications, lectures, meta-analysis, and the news articles. Okay. Here I will choose clinical trials. For the text availability, you may choose between three options. It's either available as an abstract only, or available as a free full text, or you may choose the papers that are either available as a free full text, or you can purchase the full text here. You may also filter by the reader comments and trending articles on the PubMed. Here you can use the publication dates to filter the publications by the dates. For example, 5 years gives you the publications within the last 5 years. I will choose it. I will choose a free full text. Here the species gives you both options to either choose humans or animals or both of them. I will choose humans only. Here for the languages, I will choose English, which is the language I understand. If you can speak in other language, or you can read the articles and scientific publications in other language, you may choose it here under the customize option. You will find Arabic, Chinese, Turkish, Bulgarian. Nearly every language in the world is present here. Here for the gender, I will choose the gender of my subjects as either males or females, or I may leave it blank. For the subjects, I may choose conditions related to the subject. For example, toxicology or veterinary science or systematic reviews only, or the history of medicine, topics about the history of medicine, topics about dietary supplements, topics about complementary medicine. For me personally here, I will choose dietary supplements. And then for the journal category, you may choose the journal category you want. For example, Medline for medical journals, dental journals, the core clinical journals, and the nursing journals. For For the ages, you may choose infants, adults, or children. I will choose adults. You may also choose geriatrics, as you can see below. As you see here, it minimizes the results from more than 360,000 to 333, which is very useful when it comes to tracking, skimming, and scanning through a small number of research, which I will use some of them after skimming through them to contain or cite in my publication. Let's move on to the next thing, which is PubChem. PubChem is a search method used to identify compounds. You can identify compound by one of three identifiers. One of them is its biological activity. One is the depositor provided. The third one is the unique chemical structure. You can search for a compound in PubChem using any of the following identifiers. Let's move on to Bookshelf. Bookshelf allows you to search for books that contain your search keywords. You can choose the book information about the author, the international standard book number, and you can choose the top related chapters in each book that contain your search keywords. You can even open the chapter and browse the contained data until you find the information you need. Function. The plus is one of the functions that's most widely used in the Ncbi. It's composed of two functions. The first is the nucleotide blast, which searches for a DNA sequence and retrieve homologous genes from the database. You can enter the nucleotide sequence and find similar nucleotide sequences recorded before. Or the protein blast, which is used to retrieve similar protein from the database. You can write an amino acid sequence and find similar proteins that was previously recorded in the database. There is also another function that you can convert the nucleotide blast to a protein or the protein blast to a nucleotide, which is called transverse blast. If you went through the Ncbi and clicked on the word blast, it will open. Now I will show you how to use the blast function. I will click on blast. As you can see, blast stands for the basic local alignment search tool, which means finding regions of similarity between biological sequences. And here you may find some specialized searches within blast function. One of them is a smart blast, which finds proteins highly similar to your query. The other one is a primer blast, which designs primers specific to your PCR template. The global align compares two sequences across their entire span. The CD search, which finds conserved domains in your sequence. The Geo, which finds matched gene expression profiles. The immunoglobulin blast searches immunoglobulin and T-cell receptor sequences. The VEC screen searches the sequences for vector contamination. The CD-ART finds sequences with similar conserved domain. The MOL blast established taxonomy for uncultured or environmental sequence. The bioassay, as we have targeted before in PubChem, searches for protein or nucleotide in PubChem bioassay. Multiple alignment align sequences using domains and protein constraints. Targeted loci search markers for the phylogenetic analysis. Now simply I will use the nucleotide plus function, which is simplest of all of them. I will enter a nucleotide sequence to find its similarities in the database of the Ncbi. Here I have the nucleotide sequence. I will copy it, okay, and then I will go to the plus function and enter the sequence here, as you can see. Here I may choose the search set to either human only, to either mouse only, or both of them, or others. I will choose human genome only. As you can see here, you can choose to upload your file directly instead of just copy and paste from the Word file, but I find it easy to copy and paste. Okay, then you may click here if you want to open the results in another window, but it's useless now, so I will click on the plus function to find similarities for the entered sequence. As you see, it takes somehow seconds to finish the search. After it finishes, it opens a page which contains the similarities of your sequence that you have entered. Here, the Homo sapiens insulin is the gene that I have searched for in the plus function, as it is found to be identical 100% to the sequence that I have entered in the plus function. The mRNA of the insulin is 100% complementary to the sequence. As you can see here, if the person was diseased, these lines would have not been completed in some areas, which denotes a missing part of the DNA or a changed part that's not typical or identical, and this will not give a 100% identical gene with the norm. Okay, as you can see, the lines are completed between the nucleotide I have entered and the typical one here throughout the sequence. So it is a human insulin of type variant 3. Here, as you can see, in variant 4, there is a single nucleotide that's not present in the subject that I have entered in the query. Okay, when it goes to variant 2, there is 2 nucleotides different, as you can see, and so on. A new function is the nucleotide. The nucleotide function searches the database for the nucleotide sequence of a specific gene you want to find. For example, if I read insulin, I will get the DNA sequence that codes for the insulin protein. The protein function gets you the amino acid sequence from which the protein is composed of, while the SNP function searches for the single nucleotide polymorphism in a specific gene. It finds variation between persons in the same gene. Search for and includes the insertions, deletions, mutation, microsatellites, and non-polymorphic variants. When it comes to the genome, it is the resource that organizes information on genome, including sequences, maps, chromosomes, assemblies, and annotations. For further education, the Ncbi has designed for you webinars, courses, conferences, presentations, tutorials, documentation, and they even have a YouTube channel like the one you are watching on now. Finally, I would like to say that the National Center for Biotechnology Information is a new planet of information in the era of biotechnology. Thanks for listening.